Hello everybody. I'm Niharika Gogoi and I'm a postdoc in Durham University, UK. And today I'm presenting this work, Biohybrid Sensors for Underwater Monitoring. So a little bit about the project. So this is a EU project and we are like five project partners. And I'm mainly contributing to Durham and Manchester University. And this project, RoboSynosis, as you can see from the name, it's robots in cooperation with Synosis. So it's like uh, Synosis in a way refers to a subsystem of an ecosystem, a subset of an ecosystem where different organisms are living together. And the target is that we will monitor the organisms, sweetwater organisms particularly, and also will take advantage of the bacteria for energy harvesting via NFC. And these are the different entities that we are taking into consideration in this project. And the current work that I'm presenting is a very small part of this whole project. Commercial sensors have been widely implemented for several underwater monitoring, whether it's like waste management, ocean exploration, or life form monitoring. And traditionally, pH sensors are used, or like chemical compound detection sensors are used, and temperature monitoring sensors are various sensors are used. And the problem is with that over the time we have to do calibration, and it also is labor intensive, and we have other additional problems. So in this project, RoboSynosis, what they wanted to do is like they wanted to take life forms as biological sensors and to do the behavior analysis to do the water quality monitoring. So in this talk, we are talking about mussels. So mussels are like bivalves, and they have all the organs between those valves. And a little bit about the mussels. So mussels are actually natural filter feeders. So they consume algae, phytoplanktons, and they clean the water. But they are very sensitive to the habitat. And when and this filtering trait becomes very vulnerable when the water quality changes, and that leads to their valve movement changes over the time. And so basically, mussels are also targeted here because they are densely populated and they also do not fall into the scope of animal rights. And commonly, the sensors that are used are camera. They record images and videos and do the image processing analysis, and also some filtration technique of suspended particles in water, and otherwise accelerometers and magnetometers and Hall sensors. So in this work, we have taken two types of sensor. One is Hall sensor and one is pulse sensor. So this are the position of the sensors, and the pulse sensor is basically recording the internal movement between both the valves. And the pulse sensor is also like motivated from the human heart rate sensor. Then also it's a non-invasive way, because Hall sensor, which is very common, we have to place a magnet on the upper valve or somewhere around the muscle. So that's the reason why this pulse sensor idea was proposed in this work. And this is the platform where all the monitoring was done, and it's in the University of Graz, because they only have this authority to test with the mussels there. So this is just a circuit design that was made for this sensor. And in this work, we are like currently presenting the data for 27 hours, which is like 50-hour day and 12-hour night. And then we have done some signal processing, and then, yes. So commonly what happens with the valve movement through the Hall sensor, like through the Hall sensor, we can only see the valve movement. Like when the valve is moving, the magnet senses, and then we get some data. And what happens here is that if you can see, it's very, like it does not have any pattern based on the time of the day, or it's not also periodic. So that also creates a difficulty to analyze what is creating this valve movement and on what factors these frequencies are dependent. So it's quite unclear. And that's also one reason why we then move to pulse sensor. So in the pulse, you can see that there is a clear distinction between pulse and Hall sensor in terms of the waveform. And what we concluded from this with the help of our biologist from Graz is that most of these peaks are occurring because of the filtering and the cardiac activity of the muscle. And like the whole filtering activity has different organs of it, like they have siphon which sucks the water and they do the filtration, and then they have muscles, gills, and they have different pathways of that filtration activity. So during the day when they are filtering, these additional peaks are generated, which is why they are different in terms of the time of the day also. Here is a small correlation between both the sensors, but this thing we are going to analyze it further with our biologists. But here you can see that there is a pattern when there is a sudden closing and opening of the valve. There is an additional throw which is created, but otherwise they have superimposed other small hump kind of signal. So we are still working on this, and we are also thinking about changing the frequency range of the pulse sensor, because currently we have frequency selection for more than 0.5 hertz. So when we lower it down, we are going to get more information about this. So basically, the whole idea is that pulse sensor gives more information and clearer information than Hall sensor based on the time of the day. And we are now going to correlate between these two types of sensors. So coming to an end, so in this work, we propose a multimodal sensor unit to monitor the behavior of muscles. And also, like as I told, that pulse sensor gives more idea about the movement of organs between the valves, and Hall sensor is just giving the valve movement. And this also gives an idea that freshwater mussels can be a potential biological sensors. And in this work, we are mainly trying to add additional information by monitoring the living organisms, as I said, freshwater organisms. And the other organisms of interest are Daphnia and other planktons. And then we are going to correlate with cameras, pH sensors, turbidity, dissolved oxygen, and I think electrical conductivity. So we are going to have a lot of additional information in this work. And you can also watch a YouTube video of this project. So that's all from my side. If you have any questions, I'm happy to answer.